Day plus, and thank you so much for connecting to Metric Hub. And today we are meeting once again just to push on revision amid the examinations that are just almost starting soon. We just want to work on technical questions or exam guidelines and answer series that can assist us to prepare enough towards end of term 2 examinations. So please just check that you are tuning at Metric Hub, and we are here to offer you from grade 8 to 12. Connect now, and I'll tell you now that you'll never have a problem again because we are here to support and nurture you. Without wasting any time, please note that subscribe and join membership uh, on our channel so then we can be one community. Uh, when you go to examinations, it is always important that you read instructions before answering any questions. Note that instructions are very important and ensure that you don't overwrite them. You read instructions per question, instructions per section, and of course the whole paper has got its instruction. So we have got question 1, which is multiple choice. We choose the best answer from the options that are given there. So we want to look at these questions and then technically answer them. Question 1. It needs you to identify the organelle where proteins are synthesized. Synthesis of proteins. Where is proteins synthesized? Uh, an organelle where proteins are synthesized. I'm sure if you can check this one, it is a process by which proteins are translated, which is because of the mitochondrial DNA. So that process, it actually occurs by the ribosome. It actually occurs by the ribosome. The ribosome actually reads the mRNA stage by stage, picking up the corresponding anticodons that must read, that must match it. So it is actually the ribosome where proteins are synthesized. We are saying this is produced or production. The where protein are produced, we say that that is where proteins are translated. We shall see that as we go with the life science, they call it translation, which is another term for protein synthesis or protein production. That is the same thing. It occurs because of the matching of mRNA and also the tRNA that occurs as the mRNA reads is read through the ribosome. The tissue that forms a protective outer covering in plant organs is the protective outer covering in plant organs. It is actually made by the what? By the epidermis. The epidermis, it is actually responsible for that. These are the phloem and xylem vessels are actually, they are of course responsible for transportation of water and nutrients, which we are saying they are not a protective, they don't form a protective tissue on plants. The root hairs are also not part of that. So what is the main function of the nucleus? The main function of the nucleus is actually that it is responsible for the storage. It is where genetic information is found. Genetic information of an organism, to say that the characteristics that an organism contains are stored in the nucleus. So the nucleus basically it is responsible for the storage of genetic composition of an organism. Which of the following chemical tests is used to show the presence of glucose in food? The presence of glucose in food. When we are doing food tests, glucose is tested through the Benedict solution. Benedict solution, it is used to test for glucose. Iodine, it is for starch. And then we are saying ethanol, it's an emulsion test also, but the answer which is responsible for the testing the presence of glucose is Benedict solution. Also check on Metric Hub. There is enough lecture or presentation that is also covering that subject of food tests. I have got a lot of presentations or subject videos that identifies what is being asked here. This is a diagram that is showing a certain stage in cell division. So after completion of this cell division, the nucleus of each cell, of each new cell will have, we are saying after completion of this cell division, the nucleus of each cell, each new cell we have. So now what is happening is we need to understand the stages or the process of cell division, which I need you to take note of that to say we talk about mitosis and meiosis. They are also nice, interesting subjects that if ever you can understand them very well, you will not be able to have any problems as we go. So what is happening is that after the cell division has been done, each new cell will have 4 chromosomes. Each new cell will have 4 chromosomes. What is happening is you need to understand that when we talk about cell division, when we are talking about cell division, what you have to be understood is that if it is mitosis, it actually results in cell multiplication. It is responsible for cell multiplication, that is mitosis. So it actually allows cell growth. We can talk about cell growth, which others can say cell elongation. That is, we are saying repairing of dead cells. Dead cells, when cells are dead, repairing of dead cells, it is through mitosis. It can be saying worn out cells. They are protected and they are actually replaced through the process of the process of mitosis. Of course, meiosis class, please note that it is a cell division process that is called a halving process. Halving cell division. It's a halving cell division, which you shall see that if we talk about it, it is mostly responsible for production of gametes in sexual reproduction. Production of gametes. When you are looking at this, it is the one that produces the sex cells. So therefore, it is responsible for sexual reproduction. That is one process, that's one thing that we need to understand as we describe and follow on these processes of cell division. Yeah, so we have got 46 chromosomes, and then we are saying after meiosis has occurred, it gives us 23. We have got AM chromosomes, AB, and then after meiosis we are going to have A, and we are going to have B as different cells that must be formed after cell division has occurred. So I want you to take note of this because you shall see that they are the foundation of you to understand cell division process. Which one of the following events occurs in the phase represented in the diagram above? The following events, which one of the following events occurs in the phase represented? When we look at it, it actually has to talk about, it actually identifies that the chromatin network contents to form visible chromosomes. So the contents of the chromatin network to form visible chromosomes occurs as a process of as a process of cell as the cell is preparing for cell division. So what I want you to understand is that chromatin network, it becomes thicker and visible, so therefore they can form chromosomes that we are talking about. And then the following elements are found in some compounds. Elements that are found in some compounds. Elements that are found in some compounds. So what happens is that we have got hydrogen, nitrogen, oxygen, and carbon. So which of the elements listed above are found in proteins but not in the carbohydrates and lipids? Which are found in proteins but not in carbohydrates and lipids. So if we see that, to say it is supposed to be in proteins, but it must not be forming part of what? It must not form part of the lipids and carbohydrates. Hydrogen, it is true that it can be part of that and both. It is true. Oxygen, it is oxygen, hydrogen, sorry. We are saying, if you look at this, we are saying hydrogen, it is part of proteins, and we are saying it is part of proteins, and then nitrogen is found in proteins but not in carbohydrates and lipids. So you shall see that nitrogen does not fit the oxygen we have, carbon we have. So the correct answer is, it is nitrogen. Nitrogen is not found. It's found in proteins, but of course not in lipids and other and carbohydrates. Cell that is dividing, which is also shown there. Cell division, that is also shown. And then we shall see that, like I said, cell division is very important for you to note. We have got to understand that we have got prophase 1, prophase, we have got prophase, metaphase, interphase, and telophase. So as you can see here, the question is saying which phase is shown in the diagram. In the diagram, you shall see that if we look at metaphase, we said that the chromosomes aligns at the equator, and then we are saying that the interphase and prophase, we talked about the condensing of the what? Condensing of the chromatin network, which we said that the cell is still preparing for division. So telophase, it identifies the process, which is again shown in the diagram, when the cell is actually now dividing to form two cells that are there. So it is D. Which of the following elements leads to goiter when it is not received in required amounts in the body? If the body does not have a required amount of it, it can cause goiter. I think you know, we talk about calcium is bone formation and strength. We talk about sodium. We talk about the idea of iodized salt. When we talk about goiter, we talk about iodized salt. I guess you remember that iodine is now supplemented in the salt that is being used. So we've got iodized salt, which is used as a supplement for what? Which is used as a supplement for iodine. So it is actually to say if it is not enough, that can result in, of course, goiter that we are looking at. It can end up causing goiter if if the body does not have enough iodine. Goiter can be formed or can be a problem that will result there. So we give the biological terms in each of the following statements or descriptions. Loss of water in the form of vapor through the aerial parts of the plant. Water, when it is lost from a plant, it is called transpiration. That is called transpiration. Transpiration is the loss of water vapor from plants alone. If it involved plants and water bodies, therefore it's called evapotranspiration, which is involving evaporation and transpiration. Opening through which the spinal cord leaves the skull is called the foramen magnum. I guess you remember to say if you are going to, I'm saying in our next classes where we are going to talk about evolution, you shall understand that you are going to see that we talk about the skeletal representation of the anatomy of the different primates and also the Homo sapiens sapien. So foramen magnum, it is actually an opening at the base of the skull which attaches the head to the what? To the spinal cord, which of course it leads through to the skull. So it's called foramen magnum, which is gonna be different according to the type of an organism, to say a bipedal or a quadruped, which you shall see that on a bi it is on the center and more forward. And if you talk about quadruped, it is at the back. The transparent covering of the epidermis of the leaf, that is called the cuticle. I guess if you can remember very well and refer to the structure of the leaf, the cuticle is actually the transparent covering of the leaf. Building blocks of proteins. Proteins are made up of amino acids. These are the building blocks of proteins. Proteins are made up of amino acids. Amino acids, they form proteins. And red pigment found in the blood, that is what you call homoglobin. Homoglobin. The pigmentation, the red pigmentation, that is homoglobin. Membrane around the vacuum, a membrane around the vacuum, that is also called the vascular membrane. We can just call it a vascular membrane. It's called vascular membrane. Vascular membrane is a membrane that is around the vacuum, or others can say it's called a tonoplast. It's called a tonoplasty. A pore in the epidermis of the leaf surrounded by two guard cells, that is called the stomata. The opening through which the leaf breathes with is called the stomata, or we can say a stoma, as to say the opening on the leaf through which gaseous exchange takes place. It is called the stoma or the stomata. and then the movement of gas molecules from a region of higher concentration to lower concentration. That defines, we can define that as what? As diffusion. The general movement of molecules from a region of higher concentration to region of lower concentration through because of the concentration gradient. That is called diffusion. You shall see that we talk about the movement of particles. Where they are highly concentrated, they will move to where they are less. So from where they are too high, they move there, and then they go to where there are not so much of them. So it's called diffusion. A concentration gradient affects that. The part of human skeleton made up of arms, legs, shoulder, girdle, and pelvic girdle, that is called the appendicular skeleton. It's called the appendicular. It's called the appendicular skeleton. Appendicular skeleton is called appendicular skeleton. And then we have to see that it applies to A or B or none. We need to see that it applies. Where does it apply? So you shall see that question 1.1.3, like I'm saying, it has been repeated again, was answered on Metric Hub again. Let's just refer, and you shall see that it is, of course, answered there. It is answered also at Metric Hub there. The diagram below shows two organelles found in cells. Organelles that are found in cells. So we are given that name the organelle which is X and Y. The X and Y organelle. So we can see that X is actually the mitochondria. X is the mitochondria. And Y, of course, you can see that we are saying, as you can see, that that is the chloroplast. It identifies the chloroplast. It identifies the chloroplast, mitochondria, and the chloroplast. And then which organelle is found in plant cells? In plant cells, obviously we talk about the chloroplast. The chloroplast DNA chloroplast is found in plant cells. Y is found in plant cells class. And then a plastid, which we say that it is also found in what? In Y, is known as the powerhouse of the cell. The powerhouse of the cell, it is actually the mitochondria. The mitochondria, which is also X. X identifies the powerhouse of the cell, the mitochondria. Identify parts numbered 1, 2, 3, 4. 1, 2, 3, 4. If we look on these structures, 1, 2, 3, 4. If you see 1, it identifies the stroma. And then 2, it actually identifies the granum. We have got 3. Of course, you see that 3, it identifies the starch granule. It identifies the starch granule. And then, of course, 4, I was defining it last time again on the channel, the crest. The crest is, of course, identified by 4. So you shall see that this is 1, this is 2, of course 3, and then 4. Just please be understand that you can also be asked to give the different functions and how they work. Name the metabolic process associated with X. The metabolic process that is associated with the mitochondria. It's actually, the mitochondria is actually promoting cellular respiration. The mitochondria is associated with cellular respiration. Cellular respiration is actually facilitated through cellular respiration. And of course, Y is the chloroplast. That is the one that facilitates the production of plant food, photosynthesis. Photosynthesis occurs through the chloroplast. That is where plant food is manufactured. And then the diagram below, we are to answer various questions. Some cells of a root of a plant. I am saying that quite a lot of times you shall see that in the beginning of the chapter of the term, actually, we answer such questions. So, we need to know the basic structure of a root, of the stem, and also of the leaf. I also did explain them on Metric Hub, which I guess you have been following enough. Identify cell A and B. Identify cell A and B. Cell A, it actually identifies that is the root here. That's the root here. And then you see B, it actually shows the cortex. B, it actually shows the cortex. And then stage 2 adaptations of cell A that will enable it to absorb water. 1.5 adaptation of the root here to absorb water. Adaptation of root here to absorb water. How is the root here adapted to absorb water? So you can see that the root here, it has a permeable cell. A permeable cell. A cell, if we say it is permeable, it means that it allows water to pass through. Is a permeable cell. That is 1, 2, How is the root here? It actually has got, it is actually having a thin cell, a thin cell. It is also facilitating the absorption of water, which of course is supported by, it also have finger-like projections, which are increasing the surface area for water absorption. Finger-like projections. It has got finger-like projections, which are elongated to allow absorption of water, to increase water absorption, to increase water absorption. That is the adaptation of the root here to perform or to allow or to increase water absorption. 1.5.3. In which plant organ is the above diagram found? Obviously, like I said, that this, in which plant organ is this plant, in which plant organ is this found? You shall see that, as you see, we talk about the root here. The root here are obviously found in the what? This is found in the root, or it is found on the root. It is the root where it is found. And then what is the pathway along which water moves from A to B, from cell A to B, the pathway through which water moves from A to B. From A to B, as you can see, these are connected, or they are just interconnected, which you shall see that they also have some vascular spaces that they have. So there is, of course, one. I want you to note that 1.5.4, there is, of course, you need to understand that water, it moves cell to cell via the walls. It moves from cell to cell via the cell wall. The cell walls through the intercellular air spaces. The intercellular through the intercellular air spaces. So now, they actually move, water moves from cell to cell via the cell walls, which of course it can also be through the intercellular air spaces, which of course is promoted by diffusion, by osmosis, promoted by the process of osmosis. Osmosis, I'm sure you see that it's also another process through which water moves from a region of higher concentration to a region of lower concentration. That is, of course, the water movement support process that actually enables water to move. So the intercellular spaces, air spaces, are, of course, promoted or supported by osmosis. We can go to the next question, which is a learner set up the following apparatus to investigate how temperature affects transpiration. We want to understand how temperature affects transpiration. We say loss of water from plants. Loss of water from plants is what we call transpiration. Loss of water from plants. So now we are given a cut shot, and then we have got a reservoir. We have got air bubbles, capillary tube, and beaker of water that are shown here. So the question is saying, what is the name of the apparatus shown above? This also, I have some questions that some experiments which I have shown you, which I've been talking about the potometer. The potometer, which is being shown here, it is actually used to investigate, or we can use it to check and prove temperature's effect on transpiration. So it is a potometer. State the aim of the above experiment. The aim is actually, we are going to see the speed of movement of the air bubbles. We want to say that if the air bubbles will move, and then we determine how temperature is affecting what investigate how temperature affects transpiration. We are investigating how temperature affects transpiration. So is to determine the effect of temperature on the rate of transpiration. To determine the effects of temperature on evapotranspiration. It actually identifies the effects of temperature on not on transpiration. So the effects of temperature on transpiration is what we are supposed to determine or to investigate or to prove. If temperature is affecting is affecting transpiration, so that's the experiment that is being done, and then predict what would happen to the speed of movement of the air bubbles if Vaseline was supplied to the ventral surface of the leaf. If Vaseline was supplied, so in terms of that, it actually, so to say that Vaseline, if it is applied on the ventral surface of the leaf, so now we need to understand that we can see that Vaseline actually will stop or prevent transpiration of the ventral surfaces, whereby the stomatas are actually blocked. I'm sure you see that some other advert, which is quite a lot being shown on the effect of Vaseline, where you see another leaf shrinking and the one with Vaseline is blossoming. So Vaseline will stop or prevent Vaseline will stop or it will prevent, it will stop or prevent, uh, it will prevent transpiration. It will prevent transpiration on the ventral surface of the leaf. It will actually reduce, it will stop or prevent transpiration on the ventral surfaces of the leaf, of course, since stomata will be blocked. Blocking the stomata. The stomata will be blocked and hence transpiration will be blocked. And then the question is saying, obviously, we also skipped to say what would happen to the speed of the movement of the air. The speed of the air, obviously the speed of the air, it will be reduced there. That is the first question that we were supposed to answer. The speed of the air will be reduced. Speed will be reduced. It will be very low. It will be reduced. That is, the speed will be reduced, and therefore this one to say explain your answer here is what we have already explained, that the Vaseline will obviously prevent the transpiration. It will actually, Vaseline will stop or prevent transpiration. And of course, you see, the stomata of the leaf will be blocked. That is the explanation that we are expected of on the question to say why should the air slow down. It's because the stomatas are blocked. So the question 2.41 is that we have written in red is the answer that we have written in red there. 2.15, why would these apparatus be allowed to stand before starting the experiment? Why are they allowed to stand before starting the experiment? This can actually enable a transpiration to start, to allow transpiration to start. We can do that to allow transpiration to start. That can be the reason of causing also to allow the leaf to actually be get used to the environment or temperature. Now it's to get used to the temperature. So we must allow it to get used to the temperature, which we can say, of course, the environment as well. Which we call the environment, to allow it to be used to the temperature or to allow it to be used to the what? To the environment. And then explain 2 precautionary measures that should be taken to make the results of the experiment more correct or more reliable. In order to make sure that the results are reliable, precautionary measures must be put in place. One, we need to, of course, to make sure that the apparatus are airtight, as we have seen there. One, ensure that the apparatus are airtight. Are airtight. No air is allowed to enter. That the apparatus are airtight, so that we can use, of course, Vaseline and Vaseline can be used to tightly close any gaps. To tightly close any gaps. Tightly close any gaps. We can also maybe understand that they set up apparatus underwater to prevent air bubbles from entering. Air bubbles must not be allowed to enter. So apparatus must be set up apparatus set up apparatus underwater. I think I also once explained this. Underwater, set up apparatus underwater to prevent air bubbles from entering. Air bubbles to prevent air bubbles from entering. That must be done. We can also ensure that dry the leaves of the shoot. Drying the leaves of the shoot. The shoot that is being used, the leaves must be dry. You see the leaves, these leaves, they must be dry in order to ensure that the results are reliable. And then we also need to cut the shoot from a plant underwater. The shoot must be cut underwater also to prevent air from entering. Must be cut underwater to avoid air from entering. Air must not enter, so the chute must be cut underwater to ensure that there is no air entering. And the xylem vessels must not be cut. Xylem vessels must not be cut in order to allow to allow water movement. So the xylem vessels must remain intact in order for water to move. So these are precautionary measures that can be put in place to make sure that the results are correct. And then different phases of a particular process that is being used that are being shown here. Different phases of a particular process. Different phases of a particular processing that are shown. So the question is saying, name the process represented by the diagrams above. The diagram it identifies mitosis. It identifies mitosis and then identify phases A to D. A to D. A is anaphase. And then you can see B is gonna, it also shows interphase. And then C can also be defined by prophase. And then finally, we see that the alignment of the homologous chromosomes on the equator, it also shows the metaphase. Yeah. So state 3 reasons why is the process of mitosis important. The importance of mitosis 2.2.3. Importance of mitosis. Importance of mitosis. Mitosis, I already mentioned it at first when I was cutting through it actually repairs damaged tissues. It repairs damaged, worn out. When tissues are dirty or they are damaged, mitosis can actually allow that it actually promotes growth of organisms. which we can, of course, also say that are increasing the size of organisms. And then it is also good for asexual reproduction. Promote asexual promotes asexual reproduction, where we are saying that organisms can reproduce or can multiply without the use of gametes, and then list the main events that occur during phase A. During phase A, as you can see, is anaphase. So during anaphase, events that are occurring during anaphase. So the events that are occurring during anaphase, we see spindle fibers contracting. The spindle fibers, they do contract. They do contract the spindle fibers, which I was saying previously again that you need to know that spindle fibers are responsible for the for controlling the activities of the cell. So chromosomes split at the centromere. We have got chromosomes to splitting at the centromere. The chromosomes, they do split at the centromere, allowing sister chromatids are allowed to pull apart. So you shall see that the spindle fibers, when they contract, chromosomes do split at the centromere, and that allows the sister chromatids to pull apart. And then we also have, as they pull apart, they move to opposite sides of the pore. They do move to opposite sides. The opposite sides of the pore. So they move to opposite sides of the pore, which I can see that if you look here, we are saying they move to opposite sides or to the far sides of the pore. They move to the pore. So list the main events that are occurring. That is the last question that we are answering, which is 2.2.4. 2.2.4. 2.2.4. We can go to the next question. Representing different types of cells, of course. representing different types of cells. So we need to be able to understand an animal and a plant cell. That is very important, being able to understand a plant cell or distinguish a plant cell from an animal cell. So identify the type of cells represented by diagram 1 and 2. So diagram 1, this is a plant cell. And of course, it's animal cell. Plant and animal cell. And then give the number and the name of the organelle which serves as a storage site for water and minerals. Water and minerals are actually stored in the vacuum. So vacuum, the vacuum is actually for water storage, site for water and minerals. It is actually the vacuum which is the vacuum, and then it's G. So it is the letter, give the number and name. It is G, which is the vacuum. And then provide labels for parts A, B, C. Parts A, B, C. If we look at part A, it actually shows the cell membrane. A shows the cell membrane. B, it obviously shows the nucleus. It actually shows the nucleus. B is the nucleus, which can also be termed as the nuclear pore. It can also be nucleus pore. And then C, it actually shows the cell wall. That is C. The cell wall is actually C, which you can see there is the cell wall. And then state whether part D is a permeable, impermeable, or selectively permeable membrane. Give 2 reasons for your answer. Part D, it is actually the part D, we say the part D, which is the vacuum. The vacuum is actually selective membrane. It actually is called semi-permeable. In other terms, it's called semi-permeable. So it is actually part D. Uh, it is selective. It is actually it is selectively permeable. It is selectively permeable, which I want you to understand that it actually allows water and mineral salts to pass through, and of course avoids and prevent large molecules to end. It allows passage of water and mineral salts. It allows water and mineral salts to pass through. Water and mineral salts can pass but prevent large molecules from entering. It actually prevents larger molecules. So I'm saying that you should understand that it's called semi-permeable membrane. Certain molecules can pass and others cannot. Therefore, it is called semi-permeable. And then a plant cell in a photograph measures 15 millimeters across. A plant cell, it measures 15 millimeters across. If the actual size of the cell is 0.015 millimeters, what is the magnification of this photo? The magnification, which is, of course, is the actual. When we are talking about magnification, we are saying how has it been exaggerated? How has it been increased? So what is happening is that we are saying magnification magnification is equals to image over actual is equal to image over actual, which is equals to, you shall see that we are saying actual if the photograph measures 15 millimeters across, if and the plant cell it measures 15. If the actual size of the cell is that one, what is the magnification? So it is image over actual, which is close to 15 millimeters over 0.015. That is equals to, that can give us 1,000. So the question is saying, what is the magnification? What is the magnification in the photograph? It's actually multiplied by 1,000. That is 1,000 times. That is 1,000 times is the magnification. It has been increased 1,000 times, class. That is 1,000 times. Because we are saying image over actual can give us the magnification or the exaggeration or the stretch or the enlargement. So we are given diagrams, types of plant tissue that we are given here, types of plant tissues that we are given. And then the question is saying, identify tissues B and C. Blood tissues B, you can see B is equals to the parenchyma, and then C, you can see is the collenchyma. That is it. And then draw a labeled diagram of an epithelial cell with root hairs, a label diagram of an epithelial cell with root hairs that we can draw. It needs you to say 2.2, 2.4.2 epithelial cell with root hair. Epithelial cell with root hair. So you have to see that I was showing you in the previous diagram that we see, which I mean, this is just a cross section, or it's a diagram that was already shown in part of our question that we answered before. So this can be, we have got that thing, and then we also have our other piece. That's the diagram that I'm just coming up with here. Anyway, I'm not that much of artistic, but I'll just try to give you the diagram as it should be like. Uh, that's how it might look like. It might look like that. Yeah, so this can be called the cytosol. We have got the cytosol there. Of course, you see, this can be the vacuum. We have, of course, the nucleus. We have the nucleus, and there it is the cell membrane. The cell membrane is shown, and of course, we have the cell wall, which is also shown there. And then we have got the epidermal cell. We have the epidermal cell. Epidermal cell that is also shown there, which we say the vacuole, or we can also say that it's called the cell shape. I think if you can check better back from what we have answered, that has been given also. So it's all about being able to give the heading and also label those parts as they are given there. That's what needs to be done with reference to this, to the diagram, and then give two differences between xylem vessels and phloem sieve tubes. Difference between xylem and phloem. The difference between xylem and phloem sieve. So we can have say our xylem vessels, and then we have got our phloem sieve tubes. So when it is like that, I'm sure that you know that the xylem vessels, they actually carry water and dissolved minerals. One, carry water and dissolved. They carry water and dissolved mineral salts. They carry water and dissolved mineral salts. Of course, you know the phloem, they actually carry dissolved food. They transport dissolved food, that's the phloem. And then we also need to understand that we talk about the xylem, they are also having thick cell walls. They also have thick, they also have thick walls. They have thick walls, and then the phloem are actually having thin walls. Thin walls made of cellulose. And then we have got, of course, they also have involved in upward movement. Upward movement. But if you can check on the phloem, they actually do both upward and downward movement. Do both downward and upward movement. They do both downward and upward movement is the transport dissolved food minerals, the translocation process. And then we can also mention that the fuel, the xylem vessels, they do transport from the transporting is from roots to leaves. But if you can check on the phloem, actually transport is from leaves to roots. It is actually from leaves to roots. When plant food is made in the leaves, and then it is shared through to the leaves, to the roots. So transport is from leaves to the roots, but in water is from roots to the leaves, because water uptake is happening underground, which is promoted by the roots. And then it's a table that shows nutritional information of 3 cereal packets. Each packet is a mass of 500 grams. Each packet is a mass of 500 grams. So we have got 500 grams there. We have 500 grams. We have 500 grams. So we have got protein, vitamins, fats, sodium, carbohydrates, which are all given in different measurements. So a 15-year-old girl showed the following symptoms after eating 1 serving of cereal B. After eating 1 serving of cereal B, after eating 1 serving of cereal B, a 15-year-old girl showed the symptoms that are shown below: bleeding gums, nosebleed, sore on the skin. First question is saying, name the monomer. The first question is saying, we should name the monomer for fatty, and then we are supposed to list the symptoms mentioned above, name a deficiency disease that the girl is suffering from. So I'm saying this question, if you can check it again, it's a question that is also repeating that we have answered. Please refer to the channel for more details, because we can keep on answering one and the same question. But I've just put it here for you to refer and for you just to note and check again that the question is, of course, an exam question that is also very common. So please note that you'll be given to say suitable, give one reason for your answer, by fitting nutritional values and measurements are given there. So what I'm saying is you should be able to answer these questions given that they are given in exam, or they do appear. So the passage, cells may begin to malfunction in the body. This results in cancer. Condition may be caused by cell dividing by mitosis in an uncontrolled manner. Abnormal number of cells may cause appearance of tumor. Tumors may be malignant. Radiation from the sun may cause skin cancer. Tar in cigarettes is carcinogenic and can cause lung cancer. Viruses are considered as major cause of human cancer, like cervical cancer and liver cancer. At present time, certain diet and lifestyle increase risk of cancer. Cancer can be treated in different ways. So the first question is saying, what do we call factors that cause cancer? Factors that cause cancer, it is the tar is carcinogenic and can cause lung cancer. Carcinogenic, it is carcinogenic. Carcinogenic, and then give the name of the disease caused by an uncontrolled cell division. Uncontrolled cell division, it is actually given that this condition may be caused by cells dividing by mitosis in an uncontrolled manner. This results in cancer. Cancer is this condition. Cancer is this condition that is caused by an uncontrolled cell division. And then from the passage list 3 factors that cause cancer. We are given that it is caused by tar. Tar is given there. Tar in cigarettes. Tar in cigarettes. Tar in cigarettes can cause cancer. It can also be caused by viruses are considered as a major. Viruses are also a major cause of cancer. And then it can also be caused by lifestyle. Lifestyle, at certain diet and lifestyle. Which we can also see that a diet can also cause cancer. It shows mechanisms through which enzymes function. 3.2 shows through mechanisms through which enzymes can function. So enzymes functioning. So now question is saying identify MOP. We are seeing that this is a substrate molecule. M is the substrate molecule. And then O it actually shows an enzyme. This is an enzyme that is an enzyme substrate. Of course, which is just complexing. And then P it actually shows the products. P is actually identifies the products that are produced there. So that's how it is. State the property of enzyme that illustrate that is illustrated above. What is happening here is that enzymes, they don't work haphazardly, but they are quite specific. They actually work on a specific substrate. So this is showing that it is that enzymes are substrate specific. Enzymes are substrate. They are substrate specific. We can also say that enzymes are specific to reaction. They catalyze. Enzymes are specific. They are specific to the reaction they work on. Specific to the reaction that they catalyze. And of course, we can also, enzymes, you can check here again, enzymes do not change after the reaction. Enzymes do not change. You can see it does not change after reaction. Enzymes do not change after reaction. Enzymes do not change after reaction. As you can see, the enzyme remains the same. It is quite specific. It's a lock and key. The substrate molecule fits straight to the enzyme there, and then it can be actually worked on. The enzyme substrate is actually there even after reaction. When it releases the products, it is still the same. So it does not change even after the reaction. We are given, the diagram shows different types of animal tissues that are given. First question is saying, what is a tissue? A tissue is actually a group of cells that share the same structure. A group of cells with the same structure. With the same structure, and of course are specialized to perform and perform specialized functions. Perform specialized function. That is a cell, a group of cells with the same structure and perform specialized functions. Identify tissues B and C. Identify tissues B and C. So if you can see, B is actually an epithelium. B, you can check again, that is an epithelium. And then C is actually a ciliated columnar epithelium. The ciliated is this, it has got finger-like projections is called a ciliated. Ciliated is actually called a ciliated column epithelium. Ciliated columnar epithelium. I'm saying that it is ciliated by having the fine endings which are finger-like projections there that you can see, and then tabulate the visible difference between B and C. Difference between B and C. Differences between B and C. If they say tabulating, it means you must draw a diagram. You must draw a table that can show the difference. So we can just put here a line and then we can do that. Tabulate the difference between B and C. So one, you can see that it have got no cilia. B have no cilia. It have no. B have no cilia. It have no cilia, but we can see there have cilia, this one. It have cilia that I have marked there. It have cilia, and then two, we can also see that B is cube shaped cells. As you can see the cube shaped cells, it is cube shaped. Yes, cube shaped cells. This one, it has goblet cells, which we are saying, we can say that they are long, narrow cells. Narrow. It have got long, narrow cells. And then three, you can see that no goblet cells. It has goblet cells. These are the differences that you can see when you are looking into these two cells. The difference between B and C, as you can see there, and then state the location and the function of tissue A. A, as you can see, it is attached to the bones. A is attached to the bones. A is attached to the bones. It is attached to the bones and is responsible for voluntary movements, facilitating voluntary movements. it actually facilitates voluntary movements. It is attached to the bones to facilitate voluntary movements. And then what is meant by cardiac muscle? Uh, Cardiac, it actually refers to the heart. It is cardiac, it just means the heart muscles. Cardiac is the heart class. Cardiac is the heart. The heart is basically the cardiac. So we are talking about the heart muscle is the cardiac. And then we are given cross-section through a dicotyledonous leaf, which we are supposed to, of course, to say that identify part number one. One is the cuticle. It is just repeating itself again. One is the cuticle, and then stage two functions of the part number one. One, it allows actually, it's actually one, it is a waterproof cuticle, functions of the cuticle. One, it is actually preventing water loss. It prevents water loss. That is to say that it is shining so that water cannot be lost rapidly or excessively from the plant. It is also allows light into the leaf. It allows light into the leaf. That is also because of its shininess, and then also protection of the leaf cells. It offers protection of the leaf. It provides protection to the leaf cells, and then we give a collective name for tissue 8 and 9. The tissue 8 and 9 give the collective name for tissue 8 and 9, or 8 and 9, that is called the mesophyll tissue. The mesophyll. It is actually the mesophyll tissue, number 8 and 9. And then explain two ways in which 9 is adapted to perform its function. Number 9, how is it adapted to perform its function? One, it is actually elongated deeper penetration of, it is actually elongated and it allows deeper penetration of light. Number 3.43, it allows deeper penetration of light. That is, it is elongated because of its elongation, deeper penetration of light can be also achieved. It is also have many chloroplasts. It has many chloroplasts, which of course allows absorption of sunlight. Many chloroplasts allows for photosynthesis. I'm sure that you remember to say this question is repeating. We thought that chloroplast is important for maximum absorption of light, which of course allows for photosynthesis. It has closely packed. It has closely packed cells. The closely packed cells, they actually increase surface area for absorption of light. Surface area for absorption. To increase surface area for absorption of light. And then, of course, we can also talk about it also has thin walled. It also has thin walled cells. This also allows for diffusion of substances. For diffusion of substances. It allows actually for diffusion of substances. It is also below the epidermis. It is below the epidermis. That also allows its access to light. So these are quite a lot of points that we can give you with reference to how the mesophyll is adapted to perform its function. These are the functions or the way through which we can give that it gets adapted to perform its function. Now, it's a human skeleton that is given here. Identify the part labeled A. A, you can see that this is the skull. A is a skull, and then also to which of the two main sections of the skeleton are the parts labeled. To which of the two main sections of the skeleton are the parts labeled A, D, E, and G belong to A, D, G, A. These parts we are saying, where do they belong to? Where do they belong to? These they belong to the exoskeleton. They belong to exoskeleton. They belong to exoskeleton plus the A that we have mentioned there, including the A. So, name the structure that attaches bone F to bone H and J, bone F to H and J, F to H and J, F to H and J. So these are attached together by the ligaments. The ligaments actually attaches these together and then give the letter and name of the structure that encloses the brain. The letter and name of the structure that encloses the brain, if you can see, the brain is found in here. The brain is found in here, so it is letter A and the skull. Letter A, the name is the skull. List any three functions of the human skeleton. Functions of the human skeleton. Functions of human skeleton. What are the functions of human skeleton? One, it is actually protecting delicate organs. It protects delicate, that is sensitive, fragile organs are protected by the skeleton. It also helps to store minerals. It helps to store minerals, which are also essential for the body. It allows for movement. It allows for movement. The skeleton allows for movement. For us to move, it is because of the skeleton. It actually provides a structural support. The skeleton provides structural support, and then it can also help in manufacturing of blood cells. Manufacturing of blood cells. It also helps in manufacturing of blood cells. And then lastly, I can also say that it provides muscle attachments. It provides muscles attachment. Provides muscles attachment, the skeleton. So these are various points that we can give in order to identify the functions of the skeleton. Number 6, the biological term for the longest bone of a human skeleton. This is called the femur. It's called the fem. The fem is the longest bone of a human skeleton. And then explain why in arms the pelvis of a woman is broader than that of a man. You can just understand that it has to provide support. It is it's short and broad for female in order to, it actually allows, that is to provide pregnancy support, to provide enough support during pregnancy. To provide enough support during pregnancy, and also in facilitation of easy childbirth. The child must be born. So it is because of the broad, we are saying that the pelvis of a woman is broad because it actually has to provide support in carrying of the pregnancy and also facilitation of easy childbirth during the birth process. That is also the good advantage of that. And then we are given an investigation to determine the effect of high temperature. Effect of high temperature on washing powder containing fatty digesting enzymes. Fatty digesting enzymes. The effect of high temperature on washing powder containing fatty digesting enzymes. Procedure: 2 same school white shirts were used. White shirts X and Y had the same fat stains. White shirt X was washed with detergent containing fat digesting enzyme for 3 minutes at 37 degrees, and Y was washed containing fat digesting for 3 minutes in boiling water. And then the investigation was repeated 5 times, which we said that repetition is actually done to increase the reliability of the results. So the question is saying, state the aim of the investigation. You see, this is just stated, to determine, was carried out to determine. So we are saying to determine the effect of high temperature, to determine the effect of high temperature on washing powder. On washing powder containing fatty digesting enzymes. I'm saying this is given in the question, it is given in the essay. You want to see effect of high temperature on washing powder containing fat digesting enzymes. That's the concept. Factors that were kept constant. Which factors were kept constant? You see that there was a white shirt used. So we have got the color of the shirt was constant. Color of the shirts, it was kept constant. It also have, we also had that they also had the same stain, same fat stain. Same fat stain was also found there. We also see that it was washed for 3 minutes. It was washed for 3 minutes. So same time for washing. Time for washing was also kept constant. And then we are also told about the detergent, saying type of detergent. Type of detergent was also kept constant. And then 3.63, why was the white shirt X included? X, which of course you see, we said that it is the control of the investigation. X was used as a what? It was used to actually for comparison of effect of high temperature. It serves as a control of the investigation in order to compare results. For comparison of results, for comparison of results on fat digesting enzyme. From fatty digesting enzyme, that is what is happening again there, that to compare results from fat digesting enzyme that was there in the washing powder. And then give one reason why the investigation was repeated. Repetition to increase reliability of results. Reliability to increase reliability of results. So we repeat, we use the same type of material, we maintain the environmental conditions and many other. 3 uses of enzymes apart from one given in the investigation. Uses of enzymes. Uses of enzymes. Enzymes can be used, enzymes, they can be used for tenderizing meat, use enzymes for tenderizing meat. When we are tenderizing meat, enzymes are used. They are also used in baking. Used in baking. Enzymes are also used in actually speeding up the rate of reaction. Rate of chemical reaction speed is also increased by enzymes, also for manufacturing of baby foods. Baby food is also manufactured using enzymes. You can also use it uh, in metabolic. They are also used in metabolic processes. We can give as many other uses as possible. And then the conclusion from the investigation. What conclusion can be drawn from this conclusion that can be drawn from this? We can look at the shades. The investigation was repeated. The results of the investigation are shown below. So you see, fatty stain is there. We have got a fatty stain here, which is still present on sheet Y. So conclusion, we can see that high temperature denatures fat digesting enzymes, thus preventing it from what? From removing the stains. As you can see, sheet Y was washed in detergent containing fatty digesting. It was Y was washed with fatty digesting enzyme for 3 minutes in boiling water. So the effect of boiling water affected. So we can say that fatty digesting, we are saying high fat. The conclusion that we can make here, high fatty, high temperatures, high temperature denatures destroyed fat digesting enzyme. It destroyed the fat digesting enzyme that prevents it from removing the stain. The fatty stain was therefore not removed. Removing the fatty stains. As you can see that according to the setup of the experiment, it is stated that Hawaii was washed in with detergent containing fatty digesting for 3 minutes in boiling water, but the Shet X was washed with detergent containing fatty digesting enzyme for 3 minutes at 37 degrees, not at the boiling point of the water. So that's the conclusion. So conclusion is actually drawn from what is seen. You don't decide, you don't judge, you don't think about it, but you work it, you just take it from the statements or from the results that are presented. So class, thank you so much for your time and all the best in these upcoming exams. Let's keep connected. More is coming. We have more work that is due to come on this subject. So please look forward for that. Thank you very much. Good day.